Hello, everyone, and welcome to the second seminar of Bioelevate 2026 of this year. And today we are very happy to have Rachana Acharya from University of Cambridge. And Rachana, she is a material scientist and currently a postdoctoral research associate, as I said, at the Department of Chemical Engineering and Biotechnology at the University of Cambridge. She started her academic journey as a metallurgical and material engineer at VNIT Neighbor in India and completed her master's in material science and engineering from UCLA in the US. She then moved to the Max Planck Institute for Solid-State Research in Stuttgart, Germany, where worked on organic thin film transistors for her PhD. She was awarded the Mercury Postdoctoral Fellowship in 2023 and was also awarded the Lucas Early Career Fellowship in the same year. Since 2022, she is working with Professor Roisin Ons in the research group in bioelectronic system technology, and she is currently developing versatile bioelectronic devices for understanding cell behavior and building in vitro organ models. So today we are very happy to listen to her. She is going to give us a talk titled A Versatile Biomimetic 3D Electrochemical Device for Monitoring Cell Growth and Epithelial Barrier Integrity. And before I go to Rachana, I'll just remind or just a note that if you have any question, please write them in the Q&A box, or at the end of the talk, you can raise your hand and we are going to answer all of your questions by the end of the talk. And now, with no further delay, I would say, Rachana, please, the floor is yours.
Thank you, Latifa. Thank you for organizing this amazing series and inviting me, and also for the kind introduction. So, hello everyone. Thank you so much for joining. And over the next half an hour, I'm really excited to share some of the work that I have been doing as part of my postdoc. So, I'm sure a lot of you must already be aware that currently the processes for drug development and toxicology testing are really long, really expensive, and quite inefficient. So I don't know how many of you know, but 90% of the drugs which pass the animal testing phase actually fail at the human clinical trial phase. And this is because we have a species transfer between animals to humans, and animal physiology is just very different than humans, as you can imagine. And because of this, a lot of governments and a lot of policies actually working towards phasing out animal testing and really kind of insisting on having alternate methods for these processes. So the need of the hour is one solution for this is what we call in vitro organ models, or also known as organ on chip, where what we are basically doing is replicating an organ model outside the human body so that we can continuously and dynamically monitor the biological processes. And one major need of the hour in the development of these models is how can we make them more robust and reliable? And that is something that I will be focusing on today. So what do we need to build a good in vitro organ model? The first thing is that because we are now interfacing biological systems with electronic systems, we need materials which can talk the language of both of these systems, right? So we need materials which have mixed ionic and electronic conduction. So ionic for the biological systems and electronic for the electronic systems. And a really good class of materials is conducting polymers. And kind of the workforce of this bioelectronics community is the polymer P.PSS. So as you can see in this molecule, the conjugated backbone of the P. molecule enables electronic transport, and then the ionic component of the sulfonate ion makes it possible to conduct ions as well. The next thing what we want to do is we want to make this model as biomimetic as possible. And what I mean by biomimetic is that we want to make it as representative of human physiology or human tissue as much as possible. So I'm sure most of you might be aware here that currently a lot of biology is studied on 2D surfaces, whether it's transwell inserts or flasks or petri dishes, which are typically made from glass and plastic. So these are mechanically also quite different from human tissue. But more importantly, these are two-dimensional surfaces. So what we really want, what we really are looking for is kind of more of a 3D structure so that we can build multiple like a tissue model with multiple cells involved. And the last and very most important thing, which is the crux of bioelectronics, is that we now need electronic devices which can transduce biological signals into electronic ones. So ideally, what we want is a very clear electronic signal for specific biological phenomena. And so I'm gonna go through these three things to show you how we have been incorporating these in our work. So, talking about the biology for a little bit. So, what we are mainly interested in as a research group is epithelial barriers in the human body. So, wherever you have sorry, wherever you have a tissue barrier forming, and I have some examples here on the screen, you can see that if something goes wrong with these epithelial barriers, whether it's the gut or the respiratory tract, which is the lungs, or the blood-brain barrier, it can lead to a variety of issues. So what we are really trying to replicate is this epithelial barrier so that we can test different things and the effect of different things on these barriers. So whether it's the effect of host pathogen interactions or bacteria or viruses or different drugs for that matter. So that is what we are trying to replicate. And then if you have a look, so let's talk about the epithelial barrier. If you look at the biological model, you can see that it's quite complicated with a number of cells involved and a number of processes involved. So we want to start really on a basic model and then make it as complicated as possible. So what we're going to do is we're going to look at two different cell types. So we are, of course, the main thing is the epithelial cell lining. And the second thing that we're going to incorporate in our model is the cells which sit below this epithelial cells. So they are called as fibroblasts, and they form what is called as the stromal layer. So these are the two cell types that we are going to start with to incorporate into our model. Right. So let's look at what we actually need to create this epithelial model. So one, as I mentioned, we want to create a 3D multicellular platform, which is biomimetic, so representative of the human tissue. We then need a signal transduction for cell growth and proliferation within the scaffold. So within this platform. So as cells are growing within my platform, I want to be able to record this. And third, as I mentioned, formation of this epithelial barrier on top. So here's an image of how it's going to look like in the actual model. But we're going to go through how we build this model and mainly also focus on the electronics part of it. So let's start with the first part, which is our platform. So if you look at, so these are SEM images of different connective tissue in our human body. So if you see, they have this very characteristic structure of a microporous environment. Of course, the length scales of these pores are different throughout different parts of the body, but in general, you can see that it's consisted of this microporous structure, and this is what we want to replicate. So we take our trusted polymer, P.PSS, and luckily what happens is that P.PSS is water soluble, so it forms a colloidal dispersion in water. So what we can use the triple point of water to actually create this structure. So here I'm showing you the triple point of water phase diagram. And on the right, I have a temperature profile of my fabrication process. So what we do is we fabricate these platforms using a process called freeze drying or lyophilization. And on the left, I have temperature as a function of time. So we start in the liquid phase. So P.PSS, the polymer in its aqueous dispersion. When we reduce the temperature in the system, the water part of the dispersion is actually going to freeze. So it's going to nucleate and form ice crystals. And so you're going to have a liquid to solid transition. At this point, if I reduce the pressure in my system, these ice crystals directly go from the solid phase to the vapor phase. So they sublimate out. And this is where I can increase my temperature and bring it back to room temperature. And this is the drying phase. And as you can imagine, what is going to happen is that once these ice crystals nucleate and sublimate, it's going to leave behind this porous structure of the polymer. So this is an SEM image of the conducting polymer after this freeze drying process. And you can see that we have very well kind of replicated this microporous structure of connective tissue. So that's a success. Not only that, because of the polymer, because of the mechanical properties of the polymer, we are much closer to the mechanical properties of tissue as compared to glass and plastic. So that's another win. And as I showed you the freeze drying process, what I can also do is I can change the freezing rate or the cooling rate in my freeze drying process and basically alter this nucleation and growth of crystallization of ice crystals process to basically have scaffolds of different pore sizes. So that's how we can also tune the size and the morphology of our connective tissue and of these pores if for different applications or for different organ models. And this is quite important because, as you can see, so this is an image, a confocal microscopy image of the scaffold with the cells growing in it. And you can see that when the cells start growing or start attaching, they basically line the pores of the scaffold. So it's quite important that we have a pore size which is compatible with the kind of cells we are growing, so that either the cells don't fall through or that they are just stuck at the top and don't kind of penetrate into the depth of the scaffold. So that's the idea. So now that we have our scaffold, we basically, so this is an image of the scaffold when it's out of the freeze dryer. And we typically slice this into thicknesses between 100 and 500 micrometers. And what we want to do now is to build an electronic device out of this. So some of you might know that a lot of biology is studied on these transwell inserts. The idea of that is that you can build your model on suspended in your liquid, and then you can test different things or different phenomena happening on the top side and the bottom side separately. So that's what we want to kind of fabricate. And the idea is that typically these transwell inserts have these porous membranes, which are again two-dimensional, right? So we want to replace that porous membrane with our scaffold. So here, I hope the video is visible, but what we are doing is we fabricate gold electrodes. So right now, the process of fabrication is quite limited to mechanical processes, and we assemble the devices, assemble the scaffold, contact them with these gold electrodes, and then we place them or we integrate the entire device into normal cell culture well plates, as you can see here. So that way, because these cell culture well plates are quite well designed to handle cell culture, biological cell culture. So the idea is that we make the device by contacting the scaffold with the gold electrodes and then placing them into these cell culture well plates and ready for measurement. So let's see what kind of devices we can make. So if I contact the scaffold with a gold electrode on the bottom, and then I have a counter reference electrode on top, what I can do is I can operate this device in the electrode mode of operation, and I can perform a process which is called electrochemical impedance spectroscopy. So for those of you who are not aware, what we are basically doing is we're applying a small signal AC current and then AC voltage, and then we are measuring the current, and then we are calculating the impedance of the system as a function of the frequency of the AC signal. So what you can see is that basically at high frequencies, you have this kind of plateau in the impedance, because what is happening is your system is mostly acting as a resistor. And at low frequencies, you see more of a capacitive behavior of the scaffold plus this gold electrode, and that's why you have this linearly changing impedance. So this is kind of our baseline measurement for the scaffold integrated into an electrode mode. What I can also do is, instead of connecting one gold electrode, I can connect two gold electrodes, and the electrode on top can function as my gate electrode, and I can basically turn this device into an OECT, or an organic electrochemical transistor, because now I have three terminals. And the way this device is going to work is, if I apply a positive bias on the gate, what I'm going to do is I'm going to ionize this electrolyte medium, which is going to produce these positive ions, which are going to go into the scaffold. They're going to combine with my negative sulfonate ions, and that's going to reduce the current in the system. So initially, when you have the sulfonate ions, you have electronic conductivity in the conjugated backbone of the P dot. So normally, the transistor is in on state, but then when I apply the voltage, because of this polymer chemistry, I'm going to switch off the current in the backbone. So that's how I can operate this device as a switch. So let's see how this device functions. So here what I'm showing is what is typically called as output characteristics. So as I mentioned, we are applying a positive bias on this gate electrode. So I start with a negative bias, and you can see that there is a very large current in the system. So those of you who are maybe more familiar with OECTs, you might also notice that compared to two-dimensional OECTs, which have a current of maybe microamps in the device, this device has a very, very large current in the order of milliamps. And this is really good because I now have a large signal to noise ratio in my device. And then as I increase the positive voltage on my gate, so that's where the color is getting darker, I can see that I'm reducing the current in my system until the transistor is completely switched off. So that's how I'm operating this device as a switch. Not only that, I can also perform this measurement as a function of time, right? So I can switch the gate voltage between a positive value and a negative value. And what that's going to do is that's going to switch my transistor on and then off again. And I can actually see this switching process as a function of time as well. And what this is going to do is it's going to give you an idea of the ionic mobility in the device. And we'll see how we can make use of that for our building our model. Right. So coming back to our model, we now have our 3D multicellular biomimetic platform, which is the scaffold integrated into a device, and I can have two different measurements. Sorry. So now let's see how we can have a signal for cell growth and proliferation within the scaffold. And for this, as I mentioned, the cells which we are using are called fibroblasts. So these are typically cells which sit below your epithelial barrier in any kind of in any part of your body, and they secrete a lot of proteins. They basically help the epithelial barrier to form. So it's kind of like a foundational layer for the epithelial barriers to form. So on the left, so here I'm showing a control device, which is basically no cells, just the scaffold in the OECT mode of operation. And I can see, and I'm measuring it every day, and I can see that there is some slight change here, but at the end, it again stabilizes and the device switches off. On the other hand, if I start growing my fibroblasts in my device and I check the output characteristics, I can see that I have the same effect of the transistor is no longer switching off, but this is extremely irreversible. So even at day 11, I don't have the transistor switching off anymore. So I have an increasing current in the device as days progress. And this is basically what we characterize as the characteristic electronic signal for the growth of these cells in my device. Let's have a look at the other kind of measurement that I mentioned. So here on the left, again, I have a control device and I'm measuring it as a function of time. And what I see is that basically what I'm interested in is this region where I see that the switching off behavior of the transistor is quite reproducible and consistent in a device which has no cells. But on the other hand, as the fibroblasts start growing, so the first thing that we notice is that the difference between the on current and the off current is getting lower and lower, which we also noticed with our output characteristics. But more importantly, if I look at the slope of this curve in the off region, I can see that it's getting less and less steep. So that is again what we would call a characteristic signal for the growth of my cells within my device. And let's have a look at why this is happening and kind of hypothesize what must be the mechanism of this electronic signal. So the hypothesis is that as the cells are growing, what is actually happening is that they're covering the layer of the polymer inside the scaffold, also secreting a lot of these proteins which are acting as a barrier. And then when I try to polarize my electrolyte, it's blocking the flow of my positive ions into the polymer. And which is why, if you remember, that is what is going to switch the transistor off. So the growth of the cells is actually blocking access of the cations to the polymer, which is preventing the transistor from switching off. And if I want to have a numerical value on this, what can I consider as my figure of merit? So we're going to extract two different parameters. So the first thing we're going to extract is from the output characteristics, the on-off current ratio, which is basically the ratio of the current in the on curve, so the highest current in the device, to the current in the off state of the device. So that's my on-off current ratio. And the reason I'm doing that is because the off current as a ratio to the on current is what is changing as my fibroblasts are growing. And I'm going to measure it for every device individually, right? Because otherwise, if I have varying currents, varying on currents in my device, it's not really a fair comparison to compare the off currents to kind of one particular one. So that's my first figure of merit. So the on-off current ratio as a function of time. And the second thing, because we noticed that again, there is a change in the switching off of the transistor. What I'm going to do is I'm going to extract this exponential time constant from my transient measurements, and I'm going to use that as my figure of merit. And what I want to do is I want to basically check how these two metrics change as a function of time as my cells are growing. So for that, the way we designed this experiment was that what if we have different rates of cell growth, and that way we can check how these metrics are changing for those different rates of cell growth. So basically what we're doing is that, so this is again an image, a confocal microscopy image of the whole scaffold, so that we're not accused of cherry picking and just looking at isolated areas. And I basically put down different densities of my fibroblasts on day one. So my low density is about quarter of so 125,000 cells. My medium density is 250,000 cells, and my high density is 500,000 cells, which is 500,000. And the logic is that as these cells grow, because I have varying number of cells at the beginning, they are going to grow at different rates. And I might be able to notice that in my electrical devices as well. And sure enough, if you observe them on day 5, you can see that the low density cells have grown, but not really grown in all parts of the scaffold. Whereas for the medium density and the high density, you really have the cells growing in the entire scaffold, almost covering it completely. So the color mapping is basically blue is the nuclei, red is the actin or the skeleton of the cell. And what we also notice is that collagen, which is a very popular ECM protein, is also secreted in large amounts as the cells are growing within the scaffold. So we basically have three different conditions with three different densities of the fibroblast cells, which are clearly growing at three different rates. And we want to see if we can measure this electrically as well. So starting with our first figure of merit, which is the on-off current ratio. So here, basically, I have the pink is my media control. So just before I seed the cells on my scaffold, and then as the colors get darker, I have day 1, day 3, and day 5. And what you can notice is that for the control, I actually have an increase in the on-off current ratio, but it's mostly consistent across devices. Whereas for the devices with our cells, so for the low density cells, we can see that there is a slight decrease in the on-off current ratio, which means that this switching off was impacted a little bit. But then in the medium and the high density samples where we had a large amount of cell growth, we can see that the on-off current ratio decreases quite rapidly compared to its media control. And if you notice for day 3 and day 5 for these samples, the on-off current, like the magnitude of this ratio is almost 1, which means that the on current and the off current were almost the same. So it's completely blocking access of these cations into the polymer. And so again, here are kind of zoomed in images of how these samples or how these cells look like on day 5 of their growth. Coming to the second metric, which is the time constant during the off cycle. So again, we notice a similar kind of pattern. So just as a reminder, basically, the larger the time constant means that the slower the device is in switching off. And this would be basically because of the cell growth. So we again observe a kind of similar trend where the control and the low seeding density groups have not much of a change in the time constant, but there is a large increase in this metric for the medium and high densities, which means that it's actually correlating quite well to the growth of cells within the scaffold. Right. So coming back to our model, so we have our platform, and we also have a nice measurement for cell growth and proliferation. So coming to our last goal now, which is, can we monitor epithelial barriers in this device? So if some of you must be familiar that this is actually quite a popular technique and has been done for other studies, but what we wanted to do was kind of correlate these two phenomenon. And the cells that I'm using are called MDCK2, so they're epithelial cells, and you can see that when they're growing on kind of a flat surface, they want to bind together and form this complete monolayer of a barrier. So that's kind of the goal also in the in vitro model. So let's start with our output characteristics, just like we saw with our for our fibroblasts. So just as a quick reminder, if I have a control, which is a blank scaffold, I have a consistent switching off behavior in my device. If I grow my fibroblasts, I see that this device has the switching off of the device is impacted, and I have this large off current as well. What happens when I put down my epithelial cells? So I see that on day 1 of this co-culture model, I have a decrease in my drain current compared to my fibroblasts. But then the device kind of stabilizes, and I don't have much of a change either in my on-off current ratio or much of a change in my electrical characteristics. But that's fine because we can basically use our second method, which is electrochemical impedance spectroscopy, which I'm sure all of you must be aware that has been done for multiple models and is kind of a trusted technique to look at barrier growth. To actually look at the formation of this barrier. So again, starting with a blank scaffold, we have a mostly reproducible and consistent EIS curve. Once I start growing the fibroblasts, I again see that it's mostly constant. The only change maybe I can notice is that in the capacitive behavior, which also makes sense that as the cells are growing, there's a capacitive shift in my impedance. But very interestingly, once I put down my epithelial cells, I see that there is this large change in the impedance, especially in the mid-frequency regime. And the way this functions is that we can basically model our epithelial barrier, or basically every component in this device, as an equivalent circuit element. So for example, the scaffold is capacitive and resistance. There is a resistance of the solution electrolyte. And then my cell monolayer, I'm actually modeling as a parallel resistor capacitor circuit, because the individual cells are going to act as capacitors, but the junctions, or the place where the cells are joining to form this monolayer, are going to have a resistive component. And then if I fit this measurement to this equivalent circuit, I can actually extract parameters for this resistance and use it as a metric of how strong my barrier is, or for barrier integrity. And this has been done in multiple publications. One of them I have put down here, but this is quite, as I mentioned, quite a trusted technique for measuring epithelial barrier integrity. So let's see how this actually works out. So as I mentioned, what we want to do is we want to see the effect of this epithelial barrier formation on our different fibroblast seeding densities. So here is a control. Again, I mean, this says fibroblast day 5 and co-culture day 5, but basically it had no cells. And the blue curve is after 5 days, initial 5 days, and the red curve is after another 5 days. And that's what we're going to look at across multiple devices. For the low density fibroblast cells, so which was 125,000 fibroblasts, I see that there is a slight increase in the impedance, or a slight barrier signal on the 5th day of the co-culture, or 10 days of total culture, but not really much of a change, or not really much of a barrier formation. Whereas for the 250,000 and 500,000, so for the medium and high densities, what I see is that I have this large change in this mid frequency impedance. And as you can imagine from these images, that's also because these cells are now able to form a nice complete monolayer, which is giving rise to this barrier formation. So just what I'm showing here is that, again, blue is the nucleus and red is the cell skeleton. And in green, I have stained for a tight junction protein, which is EDO1. And you can see that it forms wherever there is a nice barrier forming. I have this nice network of these tight junction proteins. And in fact, as you can imagine, that if the fibroblasts have not completely covered the scaffold because they have not completely grown, that's where you find that the epithelial cells do form barriers, but in very local regions. So this is actually an image in the same plane, right? So you can see that wherever there was space from the fibroblasts, which they did not cover the scaffold, that is where I have this barrier formation. But it's practically the epithelial and the fibroblast cells existing in the same monolayer. So the moral of the story, so to speak, is that the formation of this kind of nice stromal layer of the fibroblast is really crucial for having good epithelial barrier integrity. And if I want to test the effect of different things which are going to disrupt my barrier or going to make it stronger, I need a very robust measurement for forming this barrier. And I need a very robust system where I know that it's kind of suitable for these epithelial cells to form their barrier. And what we can do with this device is that we can measure both of these things independently, right? So using the transistor, we can measure growth of the fibroblast cells. And then if I have a good measurement, I know that my devices are ready for seeding my epithelial cells, and then I can monitor the epithelial barrier integrity with my impedance measurements. So with that, I'd like to summarize. So as I mentioned, the goals were to basically have this 3D multicellular platform, which was our scaffold, make it into an electronic device with this dual functionality, and then monitor cell growth within the scaffold and then also the barrier formation. So with that, I'd like to thank all the different people and organizations which made this work possible. And of course, all of you for your kind attention. Thank you.
Thank you very much, Rachana. That was really nice talk.
Thank you.
And by now, I would say if anyone have any question, please, it's the time now to write it in the Q&A box, or you can raise your hand and I can allow your mic. So yeah, please. Okay, we have first questions. I will allow the mic off. Just a second. Yeah, I think from Sweat, I think, if it's correct. Uh, so I have allowed the mic, so you can speak if you would like to, or if anyone else would like to start as well.
I I just wanted to
Yeah, please.
understand the difference between this and the study is electrical, and there is parallelly also microscopy done by as a parallel method. Can you just highlight what is the difference between looking at these cells as they progress from the electrical point of view, and what are the limitations of microscopy which are covered here?
Yeah, no, that's a good question. So, typically, using imaging is a very popular technique for looking at biological phenomena. So, what you basically do is you either have transfected cells or you stain for the different proteins or the cell parts, and then you use imaging as a method of checking the different phenomena. The limitation of imaging is that it's mostly an end stage thing, so you can't really have continuous monitoring as you have with electrical phenomena. So you do have live cell imaging, but it's kind of tedious, and you need labels, and you need basically to stop that process to image. So the advantage of electrical measurements is that you can have a continuous and dynamic measurement.
Thank you.
Okay, great. So we have another question from Dimitros. He's saying, may I ask you if you have some statistic with respect to device behavior? I suspect that there is some significant device variability, especially when the cell are on.
Yeah, absolutely. No, you're absolutely right. And thank you for pointing that out. That is really one of the struggles that we have had in this project. I think the main reason is that because we are currently at this stage of device fabrication where each device is manually assembled and manually kind of fabricated, that is why we have a large device to device variability. Absolutely. What we're trying to do now is kind of have a more streamlined process for making the different parts and for fabricating. So we are looking into either injection molding for the different parts or have more of a PCB structure for the electrodes. And then I think we hope that we will reduce this variability. But for now, so the data that I showed today is from 3 different biological replicates. And what we do is we basically look at the difference of whatever metric I have within the same device. So but yeah, you're absolutely right. That is one of the struggles that we faced.
Okay, great. Thanks, Rachana. I'm asking if there's any other question, and if not, maybe I can ask one.
Mhm.
Uh, what I want to ask about, like, from device architecture point of view, do you think like different monitoring system like will be preferable, for example, to do OCT or to do just an EIS measurement? So how do you see it? So it's a different device architecture with different type of things that you want to tell about the cells.
Um, Sorry, you mean that can we have different device architectures within this device itself, or what?
No, I meant like, for example, if you want to monitor the cell growth, like, would you prefer to use OCTs or just AIS? And did you do any comparison? And if there is any other thing that you want to see regarding the cells, which device architecture are you going to pick up and why and how? Yeah.
No, absolutely. So I think that is the beauty of this device, that if I want to see what is happening within the bulk of the scaffold, the OECT is a more trustworthy
Mm-hmm.
measurement. If you remember, I can also go back to
Yeah.
that slide. Just give me a second.
Sure.
Yeah. So, for example, if I
Mm-hmm.
want to monitor not just cell growth, but in my opinion, if I have a model where I want to see what's happening within the bulk of the
Yeah.
scaffold, the OECT measurement is much more is the chosen would be the chosen method, because the growth of the epithelial cells doesn't really have any kind of distinct electrical signal. Whereas if I want to monitor
Mm-hmm.
barrier growth, then the impedance measurements is the way to go, because again,
Yeah.
what is happening within the scaffold is not really impacting the impedance of the system.
Okay, makes sense. Thank you so much. Okay, so if there is no more question, maybe I will end up the seminar of today. So thank you again, Rachana. It was really nice and
Thank
insightful
you so
talk.
much for the
Thank you so
invitation.
much. Thank you so much. Highly appreciated. And stay tuned for the next Bioelevate seminar. It would be in 3 weeks. The next 3 weeks, and I'm going to send the details of the next speaker and the talk by email for all of people who registered today. So until then, I would say see you by then, and thank you again, Richana, and ciao everyone.
Thank you so much, and thank
Thank
you
you.
everyone for joining. Thank you. Bye.
Thank you. Bye.
Thank you. Thank you.
Thanks.